Today we're going to present a paper that was seminal to the field of immunology for counting populations of antigen-specific T cells. And we thought it was interesting because it'll give you a good background of how we came to where we are today with T cell biology.
The adaptive immune response is a complex network of different cells interacting with each other used for clearing infections, foreign bodies, and cancer cells. In order to achieve these goals, while minimizing damage to the rest of the body, the adaptive immune system has several mechanisms for specificity, adaptivity, discrimination, and memory. T cells play an important regulatory checkpoint in this network system, including but not limited to the requirement for their T cell receptors, or TCR, to bind with short peptide sequences produced through the processing of disease-inflicted cells presented on a major histocompatibility complex, or MHC. Evolution has designed a process that allows this adaptive immune response to maintain enough T cells to effectively expand when needed, but not more cells than needed during states of rest. The body contains small populations of naive T cells with a single type of TCR produced randomly through VDJ recombination. It is estimated there are about 10 million TCR clones in the human body at any given time, which means there will be a clone specific for any disease state peptide that may ever be encountered by a dendritic cell or for presentation to its naive partner. Once this naive T cell has bound to its TCR specific peptide, along with receiving other co-stimulatory factors, this naive population can start proliferating and begin their effector cell function. Tracking these TCR-specific populations during states of immunologic response can prove difficult due to the relatively low affinity the TCR has for its specific peptide. A cell culture method called the limiting dilution analysis that took weeks was developed to statistically estimate TCR-specific populations, but this proves very laborious and inaccurate. Our video report today summarizes a seminal published scientific report detailing a bioengineered fluorescent multimeric peptide complex used with flow cytometry to enumerate these TCR-specific populations in a faster and more accurate fashion. Next slide. The Davis research team knew that each T cell clone had more than one TCR specific to a certain peptide sequence on its membrane surface. They suspected if they could bind multiple TCRs on a single T cell to a single fluorescent complex, this would stabilize the fluorescent complex enough to count these populations using flow cytometry. So first, they conjugated a 15 amino acid AVI-tag peptide to the C-terminus of the MHC1A2 heavy chain. This heavy chain conjugate was mixed with a TCR-specific peptide and encouraged to fold to its in vivo state. This folded protein was incubated with BIR-A, which biotinylated just one lysine on the AVI-tag sequence. Then this protein was incubated with fluorescently labeled avidin, which specifically binds for biotin. This created the tetrameric MHC1 peptide presenting complex. Next slide.
The aim for this report showcases the utilization and specificity of their soluble peptide MHC1 fluorescent tetramer for counting TCR-specific populations in a fast and accurate manner with flow cytometry.
Okay, so first they prepared two different tetramers with either the HIV infection-derived GAG or polymerase peptides. Figure 1A shows where they stained a CD8 T cell clonal population specific for the POL peptide separately with either GAG or POL tetramer. They were able to count all the T cells in each stain challenge and saw very low fluorescence for the GAG staining presented by the dotted line, indicating the T cell TCRs did not bind to the tetramer, and much greater fluorescence for the POL tetramer presented by the solid line, indicating the T cell TCRs did bind. In figure 1B, this finding was correlated with the T cell clone's ability to kill B cells presenting the POL peptide, as seen in the boxed coordinates, but not B cells without the POL peptide, as seen in the circle coordinates. Next, a non-clonal population of CD8 T cells previously stimulated by autologous B cells pulsed with the GAG peptide, which caused a clonal T cell expansion, was stained with both the GAG and POL tetramers. Shown in figure 1C, all T cells counted in the POL staining represented by the dotted line were negative as observed by low fluorescence. When stained with the GAG tetramer, only 10% of the T cells counted were positive, as presented by the solid line. The remaining 90% negative cells in the GAG staining represented CD8 cells that did not have the TCR specific to the GAG peptide during stimulation. In figure 1D, this again was correlated with the T cell's ability to efficiently kill B cells presenting the GAG peptide, as shown by the boxed coordinates, but was largely attenuated when the B cells did not contain the GAG protein, as shown by the circle coordinates. Next, they showed these tetramers could be used for other infection models by preparing a tetramer for the MP peptide from flu A MHC1 processing, a flu infection. Figure 1E shows CD3 T cells previously stimulated with autologous B cells pulsed with the MP peptide for the clonal T cell expansion were stained with the MP tetramer complex. And again, about 10% of the population stained positive, as seen by the higher fluorescent peak on the x-axis. The negative and positive populations were sorted separately and were tested for their ability to kill cells pulsed with the MP peptide. Figure 1F shows the MP tetramer positive staining cells were able to kill a substantial number of these MP pulse cells, but almost no MP pulsed cells were killed by the negative staining MP tetramer T cells, as shown in figure 1F. An interesting application of this staining method, they looked at in vivo data of the HIV infected patients for GAG and POL. Again, the GAG and POL signals correlated very nicely when staining with these tetramers with their respective cytotoxicity studies, as seen to the left. For a further example of tetramer staining utility, they sorted the POL-specific T cells from the top patient who showed the highest T cell counts and quantified different effector subpopulations based on membrane phenotype staining of the immune response. And they presented how this type of staining can be used to track infections, development of the immune system during an infection cycle. Okay.
In conclusion, this study provided a more accurate and efficient method by flow cytometry to count different populations of antigen-specific T cells. Type 2, 3, and 4 hypersensitivity reactions can occur in the adaptive immune response. Antibodies can induce inflammation and cellular dysfunction. Immune complexes can form and cause the cytolytic killing of cells. The use of dissociated tetrameric protein MHC complex as an immunologic stain has proven to be useful in the study of immune mechanisms, such as those seen in HIV infection and drug development for autoimmune disorders such as Graves' disease and lupus. a form of type 2 antibody and type 3 immune complex hypersensitivity diseases, respectively.